Dear students, today we will discuss about the sexual reproduction in flowering plants, and in this chapter we will take the first part, and in this first part we will discuss about the flower and the development of microspores, means microsporogenesis. So before starting the microsporogenesis, we just look on the flower. Flower is the reproductive part of the plant. Reproductive part of plant, and this is a very fascinating structure because from this structure we will get the fruits, seeds, and the seed is called as miniature plant. Means a seed will give rise a new plant. That's why this is a very fascinating structure, and this is a reproductive part of the plant. Flower in an angiosperm is of mainly two types, that is unisexual and bisexual. Unisexual means having only one sex. Means either androecium or gynoecium. While in bisexual flower, both sexes are present, means both androecium as well as gynoecium, both are present. Just we will take an example, that is the China rose. China rose is a very popular example of bisexual flower, whereas in case of unisexual, that is the maize, papaya, etc. Now, this is the androecium, means androecium is the male sex part, and in androecium, that is this androecium has stamens, and stamen is also called as unit of androecium, and in this stamen three structure is present: anther, connective, and filament. This is anther. The point of attachment of filament to the anther is called as connective, and this stalk is called as filament. So this is a monothecous anther. And in the monothecous anther, there is two pollen sacs. Whereas in case of dithecous, this is the dithecous. Means two lobes. It has four pollen sacs. And within this pollen sac, numerous pollen grains are present. Numerous pollen grains. So these pollen sac is also called as microsporangium. Means in a dithecous anther there are four microsporangia are present. and in each microsporangium hundreds of the pollen grains will form, and these pollen grains is also called as microspores. So the development of microspore is called as microsporogenesis, and now we will see the development of microspores within the microsporangium under the process of microsporogenesis. Dear student, just you assume a picture of anther lobe. In a diathecous anther, that is, the 4 lobes are present. Basically 2 lobes, but having 4 corners, means 4 microsporangia, and in each corner only one microsporangium will form. So initially in each microsporangium a single well-celled potent cell develops, and this is called as archisporial cell. So now suppose here this is a big archisporial cell. This will divide by periclinal division and forming 2 nuclei. Periclinal division and forming 2 cells or 2 nuclei. The upper is called as parietal cell, whereas the lower is called as a sporogenous cell. Now this parietal cell will divide and redivide by the process of periclinal division and forming semicircular 3 to 4 layers and sometimes 5 layered anther wall. So this parietal cell will divide and forms 3 to 5 layered anther wall. Means this structure is like this. This is the outermost layer. Below that endothecium. This one is epidermis, and below this इस एंडोथीसियम के नीचे these two are middle layer. And below that this red layer is tapetal layer means tapetum. So tapetum is single layered, middle layer may be 1 or 2 layered, endothecium 1 layer, and epidermis 1 layered. Means basically 1, 2, 3, 4 layered structure, or maybe 5 layered protective structure of the anther wall. And within this red layer, means within the tapetum, the single sporogenous cell. This is the sporogenous cell. This sporogenous cell will also divide continuously and forms many cells. Means this sporogenous cell will divide mitotically to form a number of sporogenous cells, means it will form a sporogenous tissue. Just we assume this a single sporogenous cell. This is basically diploid 2N, and this is a big cell, potent cell, and having abundant food and cytoplasm. That's why it will undergo meiosis and forming 4 spores. But before going under meiosis, it will turn as pollen mother cell. Or microspore mother cell. So both structure is the diploid. In short, PMC and MMC. Clear? Now, this sporogenous cell undergoes, or this PMC or MMC cell undergoes meiosis, reductional division, and under the reductional division it will form 4 haploid spores. Four haploid spores or 4 haploid microspores. These spores are enclosed here in this chamber, means within the tapetum layer. Now, endodermis is a protective layer. It only gives the protection. It is the outermost layer, single layer. Below that, endothecium. This endothecium has a fibrous thickening. This endothecium has fibrous thickening. So it is a tough layer. Now the middle layer is the simple tissues of the plant cell, whereas the tapetum, this layer has also some specific behavior, and its cells are bigger in shape. Means this tapetum having bigger size cell, bigger cells, abundant cytoplasm, and binucleate. Generally this is binucleate, and in some time it may be multinucleate. And this tapetum layer having some lipid deposition, which is also called as Lewis body. And this lipid deposition has a very prominent role in the formation of enzyme. This lipid has very prominent role in the formation of enzyme. That is the outermost layer of the pollen grain. Because this is composed of sporopollen. Dear students, now just we will take the another very important matter, that at the shallow groove between the two sporangium, just let us suppose like this, some specialized cells are located, and these specialized cells located up to the level of endothecium, and these are hygroscopic in nature, and it is termed as estomium, which is hygroscopic in nature. And hygroscopic in nature means it will absorb the moisture from the atmosphere, and in the daytime the moisture will lost, and resulting it will contract and relax simultaneously, and finally it will rupture. Means this is the point of dehiscence. This is the line of dehiscence. Line of dehiscence of the microsporangia. Now, these like this, just these sporogenous cells, which is already termed as the spores. So these, when these spores will come out through this point of dehiscence, the spores are basically tetrad in organization. Means it will arrange like this. This is the tetrad. Sometimes it is isobilateral like this. Sometimes it is linear. Sometimes like T-set. T-set. This one is linear. And sometimes like decaset, means like this. Decaset. So these are the 5 different positions of the microspores which are present in a group. Means there are 4 cells. Tetrad means 4 cells. So these are the different position and shapes of the pollen grains. And in a plant, Aristolochia, fortunately all 5 positions are present. Okay, now we will take the structure of pollen grain. Dear students, this is a picture of a pollen grain. It may be smooth walled, maybe a spiny like this, and maybe like this. This is the exine and this is the entine. and it has a single nucleus. This nucleus is haploid, means only one set of the chromosome is present within this structure. Now, enzyme is composed of sporopollenin, which is a hard layer and not easily degradable. So that's why it helps in the formation of microfossils. And this sporopollenin is mainly lipid. Clear? Now in time, this is composed of pectocellulose. Pectocellulose. So it is easily degradable and it will absorb the moisture also. But the enzyme has a germ pore also, and through this germ pore the pollen tube will come out, and pollen tube is the extension of intern, not the extension of enzyme. So when this pollen grain will land on the stigma of the gynosium, the stigma will secrete the sugary fluid, which is composed of mainly fructose, and then the enzyme will rupture and pollen tube will come out through this. Suppose here is the germ pore, through this germ pore. So this is the pollen tube, and in this pollen tube the two nuclei will migrate. But before this structure, this nucleus will divide and forms. Suppose this is the smooth walled pollen grain. The two cells are formed. One is the generative cell and another is the vegetative cell. This vegetative cell has a big nucleus, that is the vegetative nucleus. It is also called as tube nucleus, and this generative cell has also half moon shaped generative nucleus. In the vegetative cell, vacuoles are also present. Now, as soon as enzyme will rupture and in time comes out through the germ pore, this generative nucleus will enter, and as well as tube nucleus, but tube nucleus will ahead to generative nucleus. This is the tube nucleus and this is the generative nucleus, GN. After the traveling of the some distance, this tube nucleus will degenerate and this generative nucleus will divide mitotically to form two nuclei. This is the first male nuclei. This is the second male nuclei. And finally, both nuclei are termed as two male gametes. The first male gamete will fuse to the egg which is present within the embryo sac of the ovule, and it will termed as zygote, from where embryo will form, whereas the future of the second male nuclei will fuse to the central cell and it will form the triploid structure endoespera. These facts we will discuss in the second or third part, and this complete structure means two male nuclei with pollen tube is also called as male gametophyte, and the formation of microspores within the microsporangium by the meiotic division of the sporogenous cell or PMC cell is called as microsporogenesis. That's all.